We're delighted to have Ann-Sophie or Anne with us today. She's long been a friend and also a friend of PhilinBioMed and the PhilinBioMed kind of work. So Ann-Sophie is assistant professor at Indiana University Bloomington in the US. She just explained that in more details. She has joint positions in the Cognitive Science program in the Department of History and Philosophy of Science. You probably know already that she has done very interesting interdisciplinary work on the history, philosophy, and neuroscience of olfaction. She published last year, in fact, at Harvard University Press, an excellent and impactful book, Smell, Sophie. I never know how to pronounce it.
You're not the only one. A friend of mine said, like, you could have done the title that's pronounceable.
It's difficult to pronounce, very beautiful to see, I must say. Very, very nice book. And the subtitle is easier. It's What the Nose Tells the Mind. And the idea is to insist on the importance of the sense of smell as a model for neuroscience and the senses in general. So yeah, I've known Ann-Sophie for quite some time. She was doing her PhD at Egenis in Exeter a few years ago, and we met in different circumstances. I think her work is very good, and I was really convinced, as I'm sure you are, that it was very much PhilinBioMed-like. And really, Ann-Sophie, we look forward to your talk right now, and thank you so much for accepting the invitation once again.
Well, thank you so much for the incredibly kind introduction and for having me. I'm glad this is recorded, so I can kind of maybe do this snippet if I give a talk again. It's like, wait, I've got just the perfect introduction. So I'll just quickly do the screen share. Let me know whether there's any issue. Can everybody see it?
It works perfectly.
Perfect. Great. So the talk I'm giving today is something I'm which is partly actually based on the work I did for the book, but I'm currently writing up an article specifically on that issue. And one of the recent developments in olfaction, which for a long time was one of the most undervalued and underexplored model systems, but there were recent developments that led to an explosion of scientific research on olfaction, as well as also public awareness of there's something interesting about smell. That also led, however, to the issue that, well, where there's suddenly attention and money, the machine learning people are not far away. And I know I'm sort of a little bit polemic, but the problem is, of course, that quite often you cannot model a system without necessarily understanding the characteristics of the system. And unfortunately, with smell, there are many misunderstandings about how this works. And a lot of the machine learning models are still built on an understanding of the visual system, or at least how we do machine learning in the visual system. And I want to show why that is actually problematic today. Let me do just, okay. So what I do today is, this is where I'm just with the right group, because I don't do the kind of normal philosophy talk where you've got a philosophical argument and then a case study, but rather actually I start with a scientific problem and the challenge that current science faces when it comes to olfaction, namely what is actually the idea behind using machine learning to understand olfaction and to figure out what's at stake here, as well as some historical background to explain why that used to be, at least for a while, a potentially understandable, useful strategy. And then to go into the conceptual philosophical analysis to go, well, what's the reasoning behind that and what's the problem with that approach? Using science to analyze the conceptual foundations of the current problem, and then to evaluate it actually why we need to look at the biology of the system, why that matters and why that gives us a different answer to what current machine learning models actually do in olfaction. So the scientific problem seems to be quite simple and straightforward, but with many things, the devil's in the detail, and that's we still don't have a good stimulus response model in olfaction. So the link between the physical stimulus space and the perceptual space is not there. And this is not a matter, I should highlight that it's not a matter of smell being seemingly subjective, what many people think, but rather it's a matter of chemical and biological complexity. So right now you cannot reduce the odor of a molecule to its molecular structure. The reason for that is you've got highly highly irregular, extremely irregular structure odor relations. The example you see here on the left, these are 4 musk molecules. And musk, of course, as you know, is like one of the key ingredients of perfumery. It's one of the most well-studied odorants in fragrance chemistry. They're completely different. There is no similarity in the structure. You've got bicyclical compounds, you've got chain structures, you have ring structures, you've got some with nitrogen. There is no commonality. And you also have the reverse, actually, that you sometimes have molecules like isosteric molecules, which have almost exactly the same stereochemistry, so geometrical shape, but they can have completely different odors. So this has been long time a problem. The next thing is it's not just simply a couple of odors that are weird, and we just have to figure out what might be weird, but it's also a matter of big data. And this is where machine learning actually comes in, because there was a recent study proposing with a mathematical model that humans can discriminate up to 1 trillion odor stimuli. That number is debated, but I think the official number at the moment is shit tons. So it's currently unknown how many really, but it's a lot. So the thing with olfaction is you've got a variety of a plethora of smells you can perceive. That's 2 of the 3 problems I want to highlight. The third one is also we seldom smell single molecules. We smell molecules in mixtures. And many odorants, many smells are actually composed of hundreds and hundreds of molecules. Coffee, for instance, has several hundred molecules, none of which individually these compounds, none of which individually smells of coffee. It's really the kind of old Aristotelian saying that the whole is more than the sum of its parts. So mixtures do behave unpredictably too. So it's kind of very hard to figure out what's going on here with the system. And I actually, as kind of a side joke, I put Calvin Klein's Eternity here, not because it's my favorite perfume, it's not, but it's full of a molecule called indole, which you see here. And indole literally smells of shit. Individually, if you take it individually, indole smells of shit. Now, that is not necessarily what you smell in the perfume, because quite often you can put something in. Of course, it changes the overall style of the perfume, but you don't necessarily smell the individual components. I also kind of like the conceptual joke that eternity literally contains shit. So that just as a one of the reasons why I do like working on olfaction is because it does allow me to swear in a professional setting. So this is, however, where the promise of machine learning actually does come in, because the most intriguing part of olfaction and why many big data people are now like, wow, this is actually a gold mine, is because it's not the fact that you just simply have a molecule and a smell, but each molecule, when it comes to ligand binding, contains about 5,000 different molecular parameters relevant to odor binding. That's a lot of data. And if you then also assume that know that, for instance, coffee has, again, hundreds of molecules, what's going on in your brain is like a lot of complexity when it comes to input of different features. And the question, of course, as well, maybe the lack of having a stimulus response model, of having a perceptual space linked to physical stimulus space, might just be a matter of complexity. If only we have the right tool. Now we have an increasing amount of computational power. Let's try this out. So this is the promise of machine learning. And this is actually a trend that especially started over the last couple of years. These are just a couple of examples to what extent you have both academic outlets. So Chemical Sense is one of the key journals in the field when it comes to chemical sensation, but also if you look at like broader outlets, I mean general outlets like science, as well as popular science and science communication. You've got Wired magazine, etc. And it's a drive towards figuring out how basically deep nose works, so to speak. You might wonder, okay, why, however? I mean, isn't smell kind of unimportant, how we used to think before the pandemic? The reason for that is, of course, money. Follow your nose or follow the money, because money never stinks, so to speak. But to crack the code in the nose actually promises billions and billions of dollars, because the fragrance industry is a billion-dollar industry and increasingly so. So this is just a trajectory about the US market, where a couple of years ago, it was over $25 billion per year. Because what you have is not just simply perfumes in which you need new molecules, but you've got body lotions, you have detergents, You have by now scented trash bags. Everything is being either descented or rescented, etc. So this is a billion-dollar industry globally. And that's where knowing, for instance, what a molecule smells like and what features are responsible for what, that's basically, this is where the money lies. And this is where the common trope comes in, which what I want to criticize today. Namely, the problem with these machine learning models is that they rest on what I call the common trope in 20th century studies on olfaction. It's this idea that we can go from molecules to perception. And you often see in psychophysical studies two kinds of data sets. So you've got on one hand, the ligands with the different kind of molecular features. And on the other hand, you've got descriptors of the smell. But these are words. Words are not necessarily perception. They're already on a cognitive level that is not the same as the sensory input and basically the sensory data that the brain is processing. What I mean by that, I come to in a moment, because it's not quite clear why a certain molecule necessarily, what makes it that we pin, for instance, a certain label to it. Could it have a different label, etc.? So words are not perception. That's what I at least want to say for now. Perfect. So that was the part actually I really wanted to highlight prior to things going berserk. It's that just in terms of the theoretical point of view, when we look at the model that underlies current machine learning approaches to olfaction, and we're getting to the empirical issues in a bit, that the issue really is the underlying notion of perception. And it's built on a specific understanding of the visual system. I should not say simply the visual system, because there's also different approaches now to how vision works. However, that has been the standard model. Like if we look at James Gibson, if we look at David Murray, there's this idea that perception is for basic stable object recognition. That's the function, the computational issue. I mean, computational for Murray, not for Gibson. And that this is achieved by like the sensory system, the receptors, and the brain extrapolating information by looking at feature extraction. There are particular features constructed together into a perceptual object. There, the function basically is to have to filter general information out of completely changing scenarios to have some stability in perception. And of course, the open question then is, what are the significant bits, both when it comes to the stimulus as well as actually the perceptual impression, and how does the sensory system represent that? And in machine learning approaches, so quite often these questions are considered irrelevant for the reason that the sensory system is treated as a black box. It's kind of this thing that you can go from input to output, and whatever is happening is not relevant because it's just the interface. And this is where I'm coming with the empirical data in terms of receptor studies to show this is quite a misleading approach. Now, you might say, well, aren't you kind of misrepresenting machine learning approaches? I wish I were. I wish I was, but unfortunately not. So this is one of the big places, Google's AI blog, where they're currently throwing a lot of money on this kind of approach. And in particular, this is actually a quote by one of the key researchers there. They actually say, based on analogous advances in deep learning for sight and sound, it should be possible to directly predict the end sensory result of an input molecule, even without knowing the intricate details of all the systems involved. So I'm not misrepresenting these guys necessarily, but this is really what they think. We can look at the molecular structure, we can make forward predictions as soon as we've got the right model. And I think this is deeply problematic for empirical reasons. That said, before I go for why this is problematic, at least for a while, this strategy did make sense. And it's a strategy you also see in many, still many papers, scientific papers being published. Because the idea was for a long time was smell as a chemical sense, and clearly the stimulus carries the information. And historically it made sense because for a long time the chemicals were the only experimentally accessible parts to study olfaction. There was no access to the sensory system itself for a long time because we didn't have the receptors until 1991. And there was also the idea like, well, if we just have the right kind of understanding of what we're looking for in these kind of molecules, then we're fine. We just need the right features, and then we can black box the system. And the first proper proposal when it comes to what we're looking for was actually by none other but Linus Pauling, who in 1946 suggested that, smell might work according to a lock and key mechanism similar to other systems. And building on that, a couple of people like, for instance, John Moore, who was one of the key pioneers in olfactory chemistry, then developed the stereochemical theory of odor. So where the idea was really that the odorant molecule works like a key to intersect with the receptor, the back then hypothetical receptor for olfaction. And you've got different groups involved in ligand binding. The first successful rule, actually, so at some point people tried to find rules to figure out, okay, what might be the relevant chemical similarity. The first successful rule was by Günther Olof, the triaxle rule for ambergris, where you've got a bicyclical compound and you've got particular atom groups in a specific place and orientation. That was in 1971. That was for a long time fairly successful, but more and more exceptions were found. One of them is actually Carinal, which scrupulously rebuts any rule of Olaf, and more of these cases, irregularities came to the fore. So that even the most successful rules had a success rate of about 50 to 60%. So this is just above chance level. And if you think about how long it takes to actually develop these molecules, this is, for instance, an illustration of that, just to emphasize what kind of money is behind that. This was in the 1990s, one page of a perfumer's notebook. This is a year of work in a perfumer's life in terms of synthesizing new molecules. And his job was seemingly simple, namely synthesize a new sandalwood molecule. Back then, there was a lot of money in sandalwood and new derivations of it. So you have a parent molecule and you twist and tweak it and try to get it as similar as possible, but just not exactly the same, but with a similar enough smell. Well, Price questions which one of all these molecules, only one of them smells of sandalwood. Which one is it? They look quite similar. And this just shows to what extent this is kind of a lot of trial and error. This is not really rational design in the classical sense. So the understanding is if we only have like a good enough algorithm, we might figure out what is the relevant feature here. Meanwhile, however, while people are still trying to do structure-odor relations, things changed in biology. And this was for a long time sort of just seen as background noise when it comes to odor and design. But this is one of the key developments. Namely, in 1991, Linda Buck and Richard Axel discovered the olfactory receptor genes. And actually, this was the discovery that catapulted olfaction from this eccentric little system into hardcore molecular biology and neuroscience, because they identified these proteins, these receptor genes, as belonging to the largest multi-gene family in most mammalian genomes. Dolphins are an exception, namely GPCRs, G-protein coupled receptors, that you also know from vision, regulation of immune responses, neurotransmitter detection, etc. So they're interesting from a molecular perspective because they are a highly diverse family in terms of the ligands they're receiving as well as the structural relations amongst themselves. Yet they have a lot of highly conserved amino acid sequence motifs. So there's an interesting topic here for molecular studies, genetic studies, and evolutionary studies that suddenly made olfaction interesting because it is a massive family. Up to 4% of most mammalian genomes are dedicated to these receptors. It's more genetic space than the immune system has, which shows you just like there's something going on with this family. And if we look at, for instance, like mice have about 1,000 of these receptors, humans have 400 to 420 of these receptor genes, which is interesting because with color perception, we have 3 color cones. With olfaction, you've got over 400 receptors. So this is just to emphasize the complexity of this system. And Buck and Axel got the Nobel Prize in 2004 for this discovery. You would think that might change things because it seems that the olfactory system is fairly simple. You've got the olfactory receptors, the sensory neurons in which they express lead directly to the olfactory bulb at the inferior frontal lobe of your brain, and then further to the piriform cortex, the olfactory cortex. That's 2 synapses straight into the brain. So you would think as soon as we've got the receptors, we should know how this works. Well, nothing happened for years and years on end. Nothing happened. And the reason for that really was that these proteins are very tricky. They're hard to stabilize. They've been only heterogeneously expressed fairly lately, a couple of years ago, by now, 2014. So for a long time, nothing happened on the biology front, and the chemists were still taking over. And there was one paper in particular in 2006 by Charles Sell, who's one of the key fragrance chemists and authority. And he's now retired. He was at Quest International in the UK. And he wrote this paper on the unpredictability of odor, which is a great paper. And he analyzes to what extent there's such an irregularity of odor and structure of molecules that the idea to just predict it is ill-conceived. Now, rather than people saying, well, maybe we need to rethink what that means, this was conceived of as a challenge, namely by a new generation that came into the field with computational methods and said, you say we can't do that, we'll show you we can. And so a lot of papers came trying to basically correlate these two data sets with a good enough algorithm. The strategy, however, of structure-odor relations, or SORS, does not apply anymore today. And that's basically the remainder of this talk. Where I want to show three issues empirically with this approach. The first one is the choice of data and data sets. The second one is the presumed link between these data sets. And the third one is the task of the olfactory system itself, which cast doubt on this kind of strategy. Starting with the data. So if we look at the kind of data that's being used, you've got a lot of papers coming out. One of the key papers was in 2011 by Alexei Koulakov and Dima Rinberg, who've been doing some really interesting studies trying to correlate particular odors to a certain olfactory impression. What they, as I already mentioned a couple of times, leave out is the biology and the psychology of the system. And you might say, well, what is the data they're actually using for this approach? They're actually using a book from 1985, Dravniek's Atlas of Odor Character Profiles, which is not a database. It is basically a book that was published to help consumer and similar communication, because quite often the context in which odorants are relevant is, for instance, cosmetic context, wine, etc. So different consumer contexts, and it's not a database. It's basically a list of descriptors for particular molecules, which were picked by Dravniek himself, which is quite an amount of work for an individual researcher. But he picked these. He did not do kind of a larger analysis on how people use descriptors for odors, but he picked them himself, and then did a couple of psychophysical studies to see how people responded to that. So what you're actually mapping is kind of like the space of the perceptual space of one person and a couple of people in a very specific cultural context of the baby boomer generation in the US. A lot of these, and this is what a couple of researchers already say, a lot of these descriptors are not applicable anymore today. They don't tell the current generation anything of what this molecule smells like. There are certain things that, like wintergreen, for instance, some people don't even know what that smells like. So it was intended as a helpful resource, but not necessarily it's not the right data to do these kind of assumptions. So we need new data, not from 1985, but what if we, however, had proper data? What if we were doing some good psychophysical studies to see how people are using these descriptors, also with a more heterogeneous population? And this was actually done recently, and also used for kind of a machine learning study, which came out a couple of years ago in 2017 by Andreas Keller and a couple of others. Came out in Science. They used new and an extended set of psychophysical data. They had also, for olfaction, I should say, a large data set. I mean, the problem with olfaction is, well, we didn't have previously, and this data set was from 2016. Why we didn't have sufficient data is also because it's a hopelessly underfunded field. And doing psychophysical studies with olfaction is, in the words of Andreas Keller, quite boring. You give people something to sniff, you ask them what they sniff, or you give them a couple of descriptors, and you do that for hours. It's not really what you think this is what I want to do when you go into science necessarily. But it was a very, very good study when they actually went through all the hardship, and they've done that. They produced some good data, and then they said, okay, let's see what we can do with this kind of with this kind of data. What is interesting is they also actually did a crowdsourcing effort, meaning they posed a challenge, the so-called dream challenge, where they said, well, find us an algorithm that connects these two data sets, and the winner is going to be part of a science of a science publication outlet. And that is the study that came out in 2017. I mean, predicting human olfactory perception from chemical features of odor molecules. That's quite a bold title. The thing is, it didn't predict anything in the strict sense how we talk about prediction. It did a classification of data, and it did a good classification of data. It did not do it prediction. But before I come to why that is, it was even picked up by really, really well-known science writers like Ed Yong, for instance. And in Ed Yong's piece, I love him as a science writer, but not a single time does he mention the receptors. So this is still conceived of, even today, as a problem of chemistry. The setup of this study was basically you've got two data sets. On the one hand, you've got the molecular features used like basically extracted from the software eDragon, and on the other hand, you had the psychophysical data set. You actually had two. You had a big data set, then the contenders were developing an algorithm to see, okay, how does that fit well, and then they were giving a second psychophysical data set to adjust basically the algorithm in order to come up with structure-odor relations. The data they were based on were 49 subjects, 476 molecules, and 19 descriptors drawn from or used from Drabnik's atlas. Sounds quite good. However, the problem with that, and that has been also criticized by people in the field, such as Avery Gilbert, who's like well-known in the field as a sensory psychologist. The one thing is there's an absence of a good psychophysical theory, because if you look at these 19 descriptors of odor quality, which you see here, there is sometimes something very, very broad like chemical, and chemical actually means nothing else than people perceive something, whatever it might be. Then you've got some which are extremely specific, like garlic, some which are more broad, like wood. And then you have something like sweet, which technically is not an odor. Sweet is a taste, it's not an odor. And there are also some things I mentioned, for instance, cross-cultural differences. Vanilla, for instance, was used as the molecule that underlies bakery. Now, you in France, and I as a German, know that bakery smells of bread, not vanilla. So this is a particular US specific term. So there's just a couple of issues when it comes to terminology. It still is also like unclear what the olfactory space actually is supposed to be here. And now I have to just briefly click you away because otherwise I can't see the quote. Avery Gilbert made a very good comment about the problem with the idea that this gives a structure-odor relation, and because it doesn't tell you anything about predicting anything from these features. Because if one wants to predict what molecules might smell of sandalwood or citrus, which is not on this list, one would have to retest all 476 molecules on another 49 sensory panelists using the new list of descriptors, then rerun the computer models on the new data set. So you have a good classification. You don't have a predictive model. Furthermore, something I also highlight earlier is that we're having words again. We're having labels. We're having no understanding of what kind of perceptual data is behind that. This is why I emphasize the difference between bakery being pinned or the label for vanilla, which we wouldn't necessarily agree with. And words are useful in an olfactory lexicon, but they occur at different levels of the olfactory categorization. And that is actually quite crucial, because smells, and this is the key part I really want to highlight in this talk, smells are not simply or just stimulus chemotropy. There's something else going on here. And the first one is, whoops, for some reason, okay. The first one is that we need to distinguish between semantic and causal objects. So you can have the same smell coming from different sources. So you can have something that smells of lobster that comes from, well, a lobster. Or you could have the same smell coming from a lobster perfume. And this is a real perfume, by the way.
Are you taking the train?
So,
I'm sorry?
Are you taking the train?
Yeah, looks.
oh, Okay, I was afraid that there was another Zoom bombing. It's like, no, not again. Well, but you've got the difference between the semantic and the causal objects. So you could have something that smells of, let's say, cheese, and it could be cheese, or you could have something that actually is an evil perfume creation by an evil perfumer who hands in their resignation. So there's already a difference between these two things. Why does it matter? Well, because there's also semantic ambiguity of many odors. You know that sometimes you've got a smell and you're like, it's familiar, but you don't know what it is. This is the so-called tip of the nose phenomenon coined by Triengen. And you can have actually the same odor associated with different perceptual labels. And this was a study by Rachel Hertz in 2001, which I really like, where she gave participants different bottles. They looked kind of identical, but they had different labels. And in one case, one was labeled Parmesan, the other vomit. Participants were adamant that these two bottles were actually not identical. They smelled different, but they had, in reality, the same mixture, a mix between butyric and isovaleric acid. And she did that with 5 different sets. And people were, in most, like over 80%, these are different, except for menthol, there was like a 50-50 chance. Because the point is not that this is an illusion or this is a deception, we can't trust our sense of smell, but butyric and isovaleric acid is part of both Parmesan and vomit. So odors are promiscuous. The same molecule can come from a variety of stuff. And in context, it has a different conceptual association. The next thing is why smells are not just simply chemistry. It's actually a phenomenon known as olfactory white, which was discovered by Noam Sobel in 2011. And olfactory white was the question like, well, is there something like white in vision, where you basically have an overload of your sensory system, too much information, everything at once, your brain can't compute, so it says, okay, I'll give this the perceptual impression white. With olfactory white, something similar occurs. So they had a mix of 30 molecules. Only condition was that there were non-overlapping causal features, but otherwise random 30 odorants, putting together at as an equal intensity, and it smelled of something you don't know from nature. So they called this artificial perception Lorax. Then they did the same with 30 other molecules, same principle, smelled the same. Then they did something with 60 molecules, same principle, smelled the same. Then they arbitrarily divided it into like 30 and 30 molecules, smelled the same. Then they did it with 90 molecules and so on. So it was always the same. So the point here was basically what happens here is that your olfactory system is so overloaded with information that, like visual white, it creates this kind of white noise thing. Okay, this is too much information, everything at once, I can't compute white. So there is such a thing as olfactory white. It's a clear olfactory impression, but it's not linkable to a particular molecular structure. Quite the contrary. It's a computational product of the brain. That leads me to the question of, well, if we look at structure-odor relations, and there's clearly a separation between this molecule and these features and this kind of perceptual image, is there sufficient reason to think that knowledge of receptors and their behavior actually affects and changes our understanding also of odor coding, the perception and what happens after? The short answer is yes. The long answer is actually really, really yes. So I'll just do the short answer here. But in order to do that, I should perhaps say a couple of things about olfaction in general, because quite often, not many people necessarily, like everybody knows vision, not so many people are familiar with the sense of smell. The key difference to vision and audition is that in vision and in audition, you've got a low dimensional stimulus. Low dimensional meaning you've got a key causal feature based on which the sensory system builds its impression. With color perception, you've got the visible spectrum. With audition, you've got pressure waves, but it's fairly linear, and you can then, of course, cut off certain ranges from the receptors, cut off certain ranges, and you can compute based on that in an additional sense. With olfaction, you don't have a low dimensional stimulus, but as I already mentioned, you have a high dimensional stimulus, meaning a seemingly simple smell, such as a rose, is not caused by one molecule, but actually a dozen of them. These are some of the molecules responsible, and each of which has 5,000 molecular parameters, potentially relevant for ligand binding. So the question is like, well, how does the brain make sense of all that molecular information and gives it the label rose? and also gives the label rose to a bunch of different roses, because of course you've got the biological cycle of a rose in the evening and in the mornings might not smell exactly the same, but you know it's a rose. Similar, you've got different species of roses that have a different molecular composition. So how do we do that? Well, the short answer is we actually don't quite know yet. That's the fun part, what I'm interested in, but that would be boring to end the talk with. So what happens here is basically you've got a really, really nice issue of, well, you've got the odorant molecules interacting with the sensory epithelium on the top of your nose, just under the cribriform plate. That's where they interact with the receptors, and each receptor, that's kind of a cool thing with the olfactory system. It's a genius engineering thing. Each sensory neuron expresses only one receptor gene. And all the neurons expressing a particular receptor gene converge in a structure called a glomerulus in the olfactory bulb at the inferior frontal lobe of your brain. And from there, here you seem to have kind of a spatial arrangement. From there, the signals further transmitted to the olfactory cortex, specifically the piriform cortex. 2 synapses straight into the cortex. Vision, 2 synapses don't even get you out of the retina. This is why people thought this should be simple. Well, it wasn't. We still don't really know what actually happens here, how smells are represented, and also, well, how does the brain actually allocate neurospace to what seems to be a non-spatial stimulus? So how, however, is that interaction already shaped here at the sensory level? What we do know from also vision, what applies to olfaction, is that you actually already have a structuring of the input at the receptor level. For vision, that's clear. For olfaction, the question is what actually happens there. So the question is not how chemistry works to understand olfaction, but how biology perceives chemistry. And here's basically why, instead of looking at kind of higher level perception and looking at this as a purely computational issue higher up in the brain, we need to start with the receptors. We need to rethink with the receptors. And this is work where I build on the work of my former laboratory by Stuart Feuerstein, who's of course also a member of your group, like like, I think he's been talking at some point to you as well. And he actually asked this very, very simple question, namely, so if we say there's chemistry and there's perception, we're kind of assuming that stimulus chemistry is coextensive with odor coding. Is that true? Because many people, and I also ask many olfactory scientists, often say, well, the receptors just perceive the chemical features, right? Well, the question is, do they perceive the same chemical features like an analytical chemist would model these molecules? And that's actually an open question, which Stuart and his laboratory actually engaged with. And it turns out currently we cannot even predict whether a molecule has a smell. So not just what molecule has what smell, but we can't even say just from the chemistry alone whether a molecule smells. That was kind of very, very nicely summarized in that quote, where it is nearly impossible to predict whether a given molecule will be odorous and what its odor quality might be from the chemical structure alone. Although all odor molecules are typically organic compounds of low molecular weight, they may be aliphatic or aromatic, may be saturated or unsaturated, and may have any of several polar functional groups. However, there are many molecules that conform to those characteristics, which are nonetheless odorless to humans and other animals. So we cannot even predict whether a molecule has a smell. And the reason for that is because we still assume that underlying odor coding is a visual model. And comparing just the stimulus between vision and olfaction, in vision I already mentioned, it's a low dimensional stimulus. So you've got a color, let's say red light, in a specific wavelength spectrum. And then you can make well-defined calculations of color combinations. So you don't have, for instance, a wavelength for pink. You've got a computation of white light minus green light. And I know the visual people often go like, well, that's kind of a simplification of the visual system. Totally agree, but then wait for the complexity in olfaction just in comparison. And what you have here is basically a high-dimensional stimulus that doesn't add up. So if you have a case like aldehydes, for instance, which you see here on the left, you have a range of different qualities depending on the length of the chain. So you've got, for instance, if you've got octanol, which has 8 carbon atoms, it has a fatty smell. You've got decanol, which has 10 carbon atoms, which is kind of citrusy, and then it gets more and more floral. But you don't have a well-defined feature combination that you could say, well, you know, you have, let's say, octanol, you add another carbon atom and you get cherry or something. This is not the same like with a visual system. And the reason for that is, as I kind of been banging on about, biology. So you've got 3 features of the receptors that explain this. One thing is you've got combinatorial coding. So you don't have a receptor that just perceives a particular feature, but you have basically each receptor can detect different molecules and also different features of different molecules. And you also have a wide tuning. So some receptors, for instance, they only respond to a narrow range of features, while others are broadly tuned and are fairly unselective compared to those. So you don't actually have an even splitting up of receptor response space, for instance. And then it gets really complicated with this. That's just like general coding mechanisms. But then you've got genetic diversity. The olfactory system is one of the most heterogeneous systems in the human, but also other mammalian genomes. So where you have individual receptor expression. So when you and I, for instance, we don't have the same receptors expressed in our nose. Every one of you has an individual receptor expression pattern in their nose. And it also changes over time throughout the lifespan of you as an individual organism, which, for instance, depends on where you are in the world. So whether you move, for instance, from Germany to South Africa, etc., you're in a different chemical environment. And as many of you know, olfaction is one of the few systems where you've got adult neurogenesis, where you've got a regeneration of the receptors, the sensory neurons, every 3 to 4 weeks. So, and this expression then changes depending on which environment you are. Besides, because of the genetic diversity, you also have different sensitivities of receptors towards chemicals. And this is, I think, really one of the strongest arguments to say why the whole structure odor approach is really problematic, because this kind of sensitivity in receptor behavior influences how you perceive the same molecule. And there are 2 examples which I think hammer the message home nicely. And one is, for instance, androstenone. Androstenone is a well-studied molecule. It's a pig pheromone, but it does more than just, you know, having pigs find each other very attractive, but it also actually smells different to different people. Some people cannot smell it at all. Some people can perceive it. To some people, it's super unpleasant because it smells kind of like urine, like not really nice. Other people find it unpleasant because it's like body odor, like sweaty. Other people find it very, very nice because it got a woody note. And other people find it very pleasant because it got a floral note. The reason for that is partly, for instance, age, partly familiarity with the smell, but also this is an increasing study in olfactory genetics that perceptual differences are caused by slight genetic differences. So same molecule, completely different categories, from urine, sweat, to floral to woody, and even different hedonic associations. So it's not even that you could say, well, that at least is stable. No, you've got the same molecule, pleasant or unpleasant, depending on how it's perceived. Another good example is, just to show how localizable these kind of genetic differences are, is cilantro or coriander. Some of you might actually not like coriander, or you know somebody when you've got a dinner, a family dinner, or you get friends over, you sometimes there's one person who doesn't like cilantro. It's like, nah, you know, it's kind of soapy and pungent. I don't know why you would put that on food. They actually perceive it different. And there's a mutation near one of the olfactory receptor genes, the OR6A2, which actually causes people to perceive the same molecule differently. So this is where I say the idea to just have the molecule in isolation of biology of the sensory system and just to attach a label is absolutely misguided. So the short answer really is that odor space involves a third list, not just ligands and descriptors that you can just throw together as a big data problem, but we need to think about the receptors. So the difference actually between, for instance, the biology and just the chemistry of the system is that the receptors, furthermore, don't respond to the same features even that an analytic chemist would highlight in their categorization of chemical similarity. And that was a study by the Firestein lab, actually 2 studies, where they did one with ketones, where they looked at, well, if we look at the chemistry classification, like how an analytic chemist would model chemical similarity of this bunch of ketones. And then they compared it with how do the olfactory receptors respond to them, and how does the similarity, like how do they actually group them as being more similar or not? You get a completely different similarity tree. So you can actually see in the figure I've got here on the right, for instance, that in the chemistry classification, odorants 3 and 4 are the closest together, while in the olfactory sensory receptors like neurons, they actually go for, well, I actually find 5 and 6 much more relevant together. So there's a difference in terms of how chemical similarity is being defined in the first place. And just to show you how different that is, chemistry, for instance, would say if you have a chemist and you give them these ketones, they would first of all go like, well, we first separate them by the size of the ring in ketones. Then they go for the composition of the ring. Is it like just one, is a bicyclic compound? Does it have one cycle? Then the subfamily, do we have a nitrogen atom there? Do we have an oxygen or sulfur atom there, etc.? And this is how they build the similarity tree. The receptors, however, the receptors, however, build on the classification of additional chemistry. Ring size doesn't even matter in the composition. Neither does actually the psychocomposition when it comes to, well, is it maybe just a bicycle compound? Is it basically just a ring? Is it a double ring? What matters is the polar surface area. So that's actually the electric charge in the 3-dimensionality of the cycle. So what you see here. That actually determines whether a neuron will accept the odorant as a ligand. And what is interesting, and that is why I say, like, well, we really need to take biology into account, that feature, polar surface area, wasn't even mentioned in that big science study by Keller et al. With machine learning about, well, predicting structure. It wasn't even mentioned. Machine learning didn't even recognize the key feature to which the olfactory receptors respond in the classification of odorants. So these are just a variety of points to say, well, the short answer is yes. The long answer is even hell yes. Like this goes even further. I cannot give the full story here, but I mentioned mixtures. And this is where we come to the question of how do things interact in combination? Because I already mentioned, like, there's no additive stimulus response model for individual odors. There's certainly none for mixtures. And this was a study by a colleague of mine at Cornell who actually figured out, like, okay, how can we, for instance, do the odor of potato chips, the perceptual image of potato chips with a couple of key odorants? And he realized, well, we can actually do rotten cabbage, potato, and toast together. But it's not just simply the set. They need to be in a specific ratio in order to create something that is like the Aristotelian, the whole is more than the sum of its parts. So you have the effect that sometimes you create a perceptual configural image out of individual components that have nothing in common otherwise. You can also have the opposite, that sometimes you add something into a mixture and it doesn't change a thing. So something else is going on here, why we need to understand odor coding really from the systems theoretical perspective. Because in the end, a natural, like an organism in the world, is constantly surrounded by hundreds of odors. So how does that work? And frankly, I actually think we need to model odor coding from the mixtures and then the individual components, not the other way around. I think there's a paradigm shift needed here. If you're interested in mixture coding, since I don't want to talk another hour here, I could, but I will not, because I think my voice at some point is like, I know it can be grating. But if you're interested, I published 2 things on mixture coding in addition to a chapter in the book. One is actually Imaging the Living Brain, where it looks at population responses to mixtures, a study also by my former lab. And another one is a chapter that will come out soon, where I wrote it together with Lu Xu, who has been doing that big study, and where she also explains the challenges of selecting the stimuli, getting the machine to work, etc. So kind of a little bit more, again, philosophy of science in action with the scientists. But to sum it up for now, the key point, okay, olfaction is not vision. Sounds true at first, but the point here was that there is no homogeneous feature extraction in the way in which current machine learning models still assume this system to work. And we need to stop black box in biology, because what constitutes chemical similarity is not a given, and it differs between the principles of organic chemistry and receptor chemistry. These are different forms of modeling chemistry. And in fact, you have, as a colleague of mine said, you could even say you've got 400 different kinds of receptor chemistry, because you've got 400 different receptor types, differently tuned, differently sensitive, potentially based on genetic differences. So if you throw in genetic variation, the idea to just do an algorithm on structure-odor relations is, I find, at best slightly too optimistic. I'm currently working on a paper of writing that up together with Lisa Lloyd under the concept of logic of research questions to show how you frame the approach really makes a difference. And I want to end at least with something, if you like this kind of topic, to point you to Avery Gilbert's blog, because he did a wonderful, very polemic, so very entertaining summary of why recent emphasis on also trying to understand perfume with machine learning. Why do we like this perfume, that perfume? It's really the old saying of garbage in, garbage out. So there's a lot of money going into the field right now when it comes to AI, but the problem is that it will not tell you neither how the system works, nor even give you proper structure-odor relations, but a minimal data fit. The question is that really what we're looking for? And with that, I want to say thank you. And I also want to say thanks to the Firestein Lab, which you see on the left, which has been doing the empirical part of the work, while I just asked them stupid questions and did my philosophical spiel around them. But I'm greatly indebted to their work. And most of you will know Stuart, so you know that he's deeply entertaining and one of the smartest persons around. So thank you for actually listening to me for that long.
Thank you so much, Ann-Sophie. That was really great. Let me clap virtually and in real life. That was great. And I really want to thank you and congratulate you for facing so bravely the interruptions that we went through. I really liked also the slide on potato chips, because it was part of the bad experience and then part of the good experience. So that was great.
Now that you mention it, sure.
that was super cool, and you really must be a great teacher in addition to the rest. So thank you. That was great. Some people had to leave, unfortunately, in part because of this delay that we got. So I'm sure there are some questions or comments. So we have roughly 20 minutes at most, but I think it's going to work in the time we have. So who has a question? Let me check if. Oh, Jan Peter has a question.
Yes, well, thank you, Thomas, and thank you, Ann-Sophie, for this great presentation. It was a good message to stop black boxing biology. I maybe would like you to invite to add, if you can and would like to, an additional layer of complexity onto the genetic diversity we'll be learning, and then related to what you alluded to, sorry, in terms of the ongoing neurogenesis and the renewal every 3 to 4 weeks of those neurons. So I was wondering if for professional noses in the perfume industry, is there anything known about how their wiring pattern can change rapidly when they go through practice sessions, things like that? Just out of curiosity.
Well, I love the question. I love that you're like, well, add another layer of complexity. You had me at, there's more to it. You're absolutely right when it comes to learning. And this is something I definitely should stress more. And now that you highlighted, there are a couple of things. One thing is, for instance, that based on learning and familiarity, it does affect receptor sensitivity. So one thing, and you don't even have to be an expert for something, but one psychophysical effect is, for instance, that just in terms of threshold tolerance, actually, the more familiar you are with an odor, the lower the threshold gets, and the more it becomes pleasant. So there are some effects when it comes to that. The next thing is, as you say, well, what about the wiring? There are less studies on olfactory experts when it comes to the receptors. However, there are actual changes in the brain. There's been a couple of studies coming out recently, for instance, by Johannes Frosnelli and Thomas Hummel and a couple of others, showing to what extent, and even within 6 weeks of training, like intense olfactory training, 20 minutes per day, you've got rather remarkable structural changes in terms of a thickening of parts of the olfactory and surrounding cortices. in 6 weeks, which is quite amazing. And then they also showed, similar to people who do mathematics, for instance, you have actually a changed activity when it comes to how much and where odors are partly processed. And there was a study, which I think is one of the best scientific studies in the last 10 years, in olfaction, definitely in the last 30 years, which came out this year by Schönhofer and Fink from the Axel lab. And what they showed, and this will have fundamental consequences for olfaction. It's like, this is, if anybody asked me what study should have been out while you're writing this book, I would have loved to have this study out. It actually emphasizes what I've been saying and banging on about the book. It's like, this just proves me right. Finally, it would have been bad otherwise. But they showed that within, like in mice, they showed that within 28 to 32 days, you've got a complete representational shift in the piriform cortex, meaning that they had mice responding to certain odors, and none of the neurons responding to that odor were the same after a month. This is so far the only primary sensory cortex where you have a representational shift, you know it in some other systems. So learning can delay that kind of shift, but it cannot prevent it. So there's a couple of things going on also with the complexity introduced by learning. There are a couple of people like John McGann, for instance, at Rutgers, who also says we need to look more in terms of top-down influences also, for instance, not just piriform to bulb, but also bulb to sensory periphery. There is work in progress. There's a lot of unpublished work, so I can't say too much there, but yes, the short answer really is, oh yes, yeah, you're pinning it down. I thank you for actually highlighting that.
Well, thanks for your answer. That's a great direction to follow. That. Thanks a lot.
Gregor.
Thank you very much for a fantastic talk, especially despite the disturbances. Thanks a lot. I was very intrigued thinking what you said at the end about the limits of AI, and was thinking about the claims about AlphaFold having solved the protein folding problem. And I think the same lessons apply here, but I wanted to hear your opinion on the next step when people talk about the structure-function relationship of proteins and predicting functions based on just the molecular structure. Do you see a connection there with the structure-odor relationships being a special instance of a more general structure-function relationship framework? And are there useful lessons for and from the Pandora box that is the notion of function debate in philosophy of biology? I would love to hear your thoughts on that.
Excellent question, because this is one where I'm also working myself my own way through the issue. Because on the one hand, I do think there's but despite me bashing machine learning in this talk, this is actually not how I see machine learning. I do think there is an utter potential here. The issue for me is the premises on which we build these models. And the problem is that olfaction has been kind of mimicked as just a chemical sense, so the biology has been excluded. If we were to take the receptors a bit more into account and also look, for instance, at these kind of differences, I'm pretty sure we could solve a couple of issues also when it comes to, let's just say, species-specific differences as well, when it comes to certain responses. I do think there's a potential here. And that can be seen, for instance, with actually success in protein folding models. So I'm not saying machine learning can't do anything good. It's just that the conceptual foundations right now are nonsensical. We're having the wrong models, so to speak, the wrong conceptual foundations. I also think where the future is right now not necessarily with throwing models at the receptors as such, and the reason for that is also a lack of empirical data in many cases. So we don't, for instance, have enough data on deorphanized receptors. Deorphanized meaning we don't really know the binding spectrum of all olfactory receptors. There are a couple which are deorphanized, and this is still ongoing. This is intense labor. Like this takes time. There's some people like Matsunami at Duke who does that now for years, and great work, but there's a lack of data when it comes to that. Where I would see a lot of promise, especially in the next couple of years, and also funding promise, is going to look at olfaction in medical contexts. So especially when it comes to bioelectronic gnosis, where you use, for instance, machine learning to figure out, can we do profiling of just like skin responses in terms of how people smell? And a colleague of mine, Andreas Mersching, who works on that, it's like, well, can we, for instance, just have something that is just even smaller than your mobile phone, which you carry with you, that records kind of a stable baseline of your skin odor, because certain skin cancers, of course, change the odor profile. So it might be a good way, and this is not not simply chemicals, but this is, again, what I'm banging on about ratio. And he's very explicit in, we need to model this more from a biological perspective, both in, for instance, using receptors plus algorithms, so he does bioelectronic noses, as well as looking at, well, how do medical detection dogs do that? So he's working with Claire Guest, who's one of the key people in the UK, with medical detection. So this is where I think a lot of things will happen in the next couple of years. With the original proteins, not yet. It's a lack of data and the wrong conceptual foundation. But at some point, I would love to see where some people might take this.
Thank you. Also,
thank you for your question, because it's like, I know I often bang on about, machine learning, they didn't solve it. Well, sometimes I'm a bit too strong in terms of polemics, so I have to not backtrack in terms of what are the arguments, but to soften it and say, I do see definitely like huge potential. Just right now, it's misguided.
The next question is by Maël.
Thank you very much, Anne. That was a great and fascinating talk. I have a question on medical cases. Back in the days when I was studying mental disorders and other diseases, actually, I was struck by the fact that in many of them, many different conditions, actually, you can see alterations of this of the smell that are quite striking. In depression, it's well known. In Alzheimer, it's also well known. In schizophrenia. And also in epilepsy. So I've encountered several patients with very weird signs and so on. And one of the questions I have still unsolved question is it, do you think we have any element to say that it can be either a bottom-up problem or a top-down problem? What I mean by that is that it could either be an alteration, molecular alteration of well, somewhere in the brain, I'd say, or even in the nose, or it could be something that involves more cognitive phenomena and impose another or some kind of other interpretation of the same stimuli. So that's roughly put, but I guess you see where I'm going.
This is a very good question. So the questions, of course, what kind of effect we're talking about, because you mentioned on the one hand Alzheimer, on the other hand epilepsy. So there's different olfactory effects. So you've got either where people lose their sense of smell, Alzheimer, Parkinson, Lewy bodies, etc., or where people actually have smell hallucinations. Epilepsy, multiple sclerosis, you've got it too. So there seem to be different issues going on. I think in some cases, it is really on the deep down molecular level when there is a degeneration. In other things, I do wonder to what extent there are definitely kind of cross effects higher up in terms of cognitive effects. And since you mentioned epilepsy, that is one of those things that why, for instance, smell hallucinations or certain things were not immediately associated with epilepsy. This has for a long, long time been too anecdotal. So quite often people were reporting things, but it was just seen as anecdotal with many smell dysfunctions. It's just anecdotal. I mean, that was why smell dysfunction was taken up as a symptom far too late in the COVID issue as well. I mean, really, really late, surprisingly late. And one thing with epilepsy in terms of cognitive effects is that quite often people have smell hallucinations, or they also have parosmia, that you've got a distorted sense of smell. So certain things suddenly smell often actually really bad. And there was one example which I found quite fascinating of a Scottish nurse who everything started to smell bad. Coffee smelled of shit, cucumber smelled of shit, everything was either urine or feces. And she didn't have seizures or anything, none of the kind of typical epilepsy symptoms. And at some point, somebody actually gave her epilepsy medicine. And she said that she noticed that her sense of smell, or that everything was kind of starting to smell normal, not by, oh yes, suddenly I smell coffee and cucumber. But the space in which she was, her room, looked different. That's odd. So that there's a different awareness of space that is somehow connected to the olfactory system. There are definitely some cognitive cross-modal effects, which I find quite fascinating. I even had a similar experience myself, not with epilepsy, but also with a change of spatial awareness through a change of smell in an otherwise habitually acquired sensory environment. So I said, like, definitely most of it will be molecular. However, I think we should also focus precisely on these kind of cross-modal interactions and the cognitive impact. I think there could be a couple of things where by training people on smells and using smell as a form of therapeutic tool, similar to Oliver Sacks used music as a music tool, to use it, for instance, in Alzheimer's to just slow down a certain decay. I think there's a lot of therapeutic potential here. It's just not very profitable for some areas of funding, let's put it that way.
Thank you very much.
Very interesting. I don't see any other questions right now. I have a question. Simon has a question. Simon has a question. Go on, Simon.
Yeah, hi, thank you. This is just in a way a follow-up anecdotal information on some of these weird cases. So I have a friend whose smell for some reason disappeared when he was young, and he can't smell anything now. So we did an experiment at home. He's my I'm living together with him at the moment. So we did an experiment where I tried to expose him to different weird things, and well, none of them really worked besides one. A particular tea, a tea I brought from Denmark, and I thought, I mean, do you have any thoughts on this, or I mean, or are there studies on people who lack smell can sort of get some senses of smell back, or something like this?
Absolutely. So actually, there are two things here. One is that you can have partial anosmia. So sometimes you're selectively anosmic to certain smells. That can be very selective, that you're just not responsive to one particular odorant. Let's say TCA, which causes corked wine. Charles Spence, the cross-modal researcher, for instance, cannot smell it, so he never has a bad bottle of wine in his life. This could, of course, be expanded, that you've got a range of smells you're not responsive to, and you might have just a minimal amount, like, for instance, your friend with tea, which I find fascinating, but you can regain the ability to smell. It's if he's mainly anosmic since like an early age, the success rate of that is debatable. But one of the key things of anosmic people, and there's been a lot of patient advocacy, especially in the last couple of years, is to help people to train their sense of smell, and you can regain it sometimes. So that's possible. It kind of sounds weird to say, well, you need to do smell training, and people look at me weirdly. It's like, you what? But it's kind of physiotherapy for your nose. And what you really do is, and you can get smell kits for that. Some people just smell whatever they have in the kitchen every day until they smell something. The more professional aspect really is, and you've got two in the UK, one is Fifth Sense, the other is Absent by Chris Kelly, who used to be anosmic, and they have smells in different concentrations. So you test every time until you get more and more sensitive to the lower concentration. The other thing is you might have a mixture. So you've got a particular odorant within a mixture of something else in different concentrations. So when do you actually, for instance, recognize, let's say, the citrus in whatever kind of overall mixtures you have? And you do that for 20 minutes per day. So this is like really not just I sniff something, but it's cognitively demanding. So it is possible, actually. Yeah.
Cool, thanks.
Well, thank you. And also good luck to your friend. I mean, you can, I like the fact that I give him strange things to smell. It's like, oh yes, I like to do that too.
And we will remember that the Danish tea has some special effects. So this is something to keep in mind. So Jonathan is next.
Yes, thank you very much. Sorry, my camera's not working properly at the moment. But first of all, great talk. It was really interesting, and it's a lot of areas, I guess, that I'm not as familiar with. This whole area of olfaction, I guess, and the science behind it. So it was really great to learn a lot. And one of the questions I had was just kind of out of curiosity, in a sense, is to, so there's a general field, I mean, getting into evolutionary medicine and Darwinian medicine, and kind of within this area, there's this notion of a mismatch and how certain organisms, ourselves included, may have evolved to develop certain traits and properties at a certain time, and that were adaptive under certain environments, and that under our current modern environments, they've become maladaptive for various reasons. And I wondered to what degree, I mean, this very sketchy version of it, but nonetheless, I wondered to what degree similar kinds of arguments or this kind of stuff has been developed with regards to olfaction, I guess, given that it might be something very difficult to determine, I guess, what it was adaptive for, and all these sort of things in an evolutionary environment. So.
I love that question. So I'm often quite skeptical about evolutionary explanations applied to smell because they often end up to be just those stories and just certain kinds of how we think smell works rather than really giving a good understanding of the biology. That being said, I don't think it's maladaptive to have this flexibility. This is when we look at what smell actually does to the organism, why I think it's so important to look at smell really as a biological sense of an organism within an environment. Our chemical environment is constantly in flux. So you're changing all the time, like environments, and with that, of course, the chemical environment. So it's very important to be flexible in your responses rather than hardwired, because many odorants come from different sources, and within different chemical sources have a different meaning. I mentioned, for instance, indole in perfume, and you have it, and I'm sorry to do that, I do that often, but I'm going to ruin coffee for you. When I mentioned coffee, for instance, it also contains about 3% of indole. Your coffee literally contains shit. Similar, you've got jasmine, which is one of my favorite flowers. Minimal amount of jasmine also is indole. And you might say, okay, but we don't smell it. That does not mean we can't detect it, in the sense that there was a study of people being able to discriminate between indole-laden jasmine and synthetic, so indole-free jasmine. They could not just simply tell the difference, but they preferred the shitty version. So there's in certain contexts, contamination is not necessarily bad, and it can also enrich a perfume or smell, as many perfumers know, actually. And I think the most important part really with smell is, what is it for? So not just simply what does it coat, but what is it for? And it's a response to an environment where it's more a matter of how much and in what combination something is, rather than the individual stimulus as such. So I often say, like, rather than looking at understanding the perceptual system as mapping the environment, which is still kind of a visual approach, I find it better to understand olfaction more as a measurement of the environment. It measures the statistical frequencies of chemical features in combination. And in some combinations, especially in certain concentration, it can, of course, then get harmful. And this is what the system responds to. But when it comes to individual odorants, there needs to be a behavioral flexibility. And I do think this also applies to other animals, not just to humans. Because if we look at most smell studies, you've got mice in these kind of little cages in a very, very environmentally restricted sense. And you stick basically a Q-tip in there with something, and the mouse is excited because something is finally happening. But then you use, for instance, fox pee, and the mouse freezes and nothing else. And so it's like, oh yeah, there's a hardwired response to certain odors. Well, then you go through New York and you see mice and you see rats, and there's constantly some predator pee, and they give a shit. I mean, they mate, they find food, they just do everything because they're constantly surrounded. So there's something, sure, there's a certain behavioral reaction to certain smells, but quite often organisms respond to multiple signals. So there needs to be a flexibility to overrule certain responses as well. And I think if we were to study olfaction more from that perspective, we would get a better perspective, both not just simply the chemistry and the biochemistry of it, but also when it comes to what you say, a better evolutionary understanding, because quite often our evolutionary understanding comes from kind of like the finished system, and then we're just reasoning back, and I find that problematic.
Great, thank you very much for that.
Thanks.
Okay, we have to start soon, but Amiriza, if you have a short question, that could work.
Yes. Do you have my voice? My internet connection is not very good. Okay, so first, thanks for the great lecture. There was so much information that I didn't have before, especially after the pandemic. We had lots and lots of questions about smells, and I didn't have any answer to. Sure, I have some now. My question may seem off topic, but we have lots of patients who are, for example, suing their surgeons after nose job or after traumatic accidents, claiming that they don't, their smells are different, or their smells are damaged or something. Do we have any objective ways nowadays to confirm that we are just relying on subjective information from patients. Can you smell this? No. Okay. Then can you smell that? No. Okay. Is there any way to just clarify if they are malingering or something like that? Thanks.
I love that question. So what you pointed is unfortunately a phenomenon that's far too widespread, but not well enough investigated. That being said, there has been research on it. So when it comes to partly, especially after nose jobs, for instance, sometimes people completely lose their sense of smell. Sometimes they develop parosmia, also with accidents, also with other things like COVID, for instance. There are different assumptions of what's been happening. Unfortunately, there's not a full understanding of it yet. Is there kind of, for instance, when basically the epithelium wires it back to the bulb, is there miswiring? One of the potential options that's being currently explored. The other thing is, well, is there some kind of misassociation? Open question. That being said, there's a way to study that. There are a couple of clinical smell tests that are used in a variety of settings, and there are two big ones to look at, well, do people, A, can they smell? B, do they smell what usually people associate with a certain mixture? There's one, and this was one of the first smell test kits by Richard Doty developed in 1984, 85. It's the University of Pennsylvania smell test identification kit. And there's another one, so this kind of a scratch and sniff test where you've got people, for instance, you've got a range of smells, 10 smells, commonly known ones where you have to, for instance, identify them with a given set of descriptors and these kind of things. A much more comprehensive test was later developed by Thomas Hummel in Dresden, the sniffing sticks, where they test with these kind of felt tips intensity, quality, and discrimination. So identification and discrimination, because these are different processes that can be differently affected and might indicate, for instance, even what kind of disorder it might be. So for instance, Alzheimer and Parkinson have different rates of decline of smell, and also certain smells seem to be declining differently. So if you can't smell peanut butter, that's not very good. That's often a sign associated with Alzheimer. If you can't smell oregano, that's often associated with Parkinson. So it's very, very interesting. Nobody knows why, but it's not so subjective. You can actually measure it. It's, for some reason, these tests are not used often enough in clinical settings. But if you're interested, Thomas Hummel is at the Technical University in Dresden. They're doing a lot of clinical research to really show we can do something here beyond just individual reports of something smells funny. And I hope there's going to be more research. And I think the pandemic really emphasized to what extent smell dysfunction is a real issue for many people, because I noticed a lot of my students, for instance, lost their sense of smell and they were miserable. They're like, nothing tastes well, like everything is just bland. There was a lack of quality of life. So quite often, many doctors, for instance, said, when I talked to anosmic patients, that quite often the doctor said, it's just the loss of quality of life. That's not so bad. And it's like, what do you mean just? Like, quality of life is the part that makes life worth living. So I think there's now at least a bit more attention to that. And I find the most interesting description of an anosmic patient I ever heard about losing their sense of smell it was as if suddenly the world was behind glass. I was disconnected from everything around me. Because in a certain way, smell, since it's a direct sense, like you touch the molecules, your receptors in the molecules do directly touch and tango. It's kind of like an extended sense of touch of the invisible molecular world around you. So you're otherwise really cut off. There's a lot of information you're not getting, even though you might not consciously process it. So even though there's variation in patients' responses, I don't think it's necessarily subjective in terms of that there's no causality behind it. If that answers your question, I mean, no.
Yes, very good. Thanks.
Thank you.
Okay, I think it's time to stop. But before stopping, I really want to thank you again, Ann-Sophie, because the talk was great. And also I found the answers to the questions very specific. And it's very impressive to see the scope of your knowledge about smell. I think that is great. And as we saw, it touches on all sorts of different questions. So thank you everyone for the questions. And thank you very much, Ann-Sophie, for the talk. I hope we can make a video without the hackers. We'll see. We'll do.
With special effects, maybe with Monty Python in between. But thank you so much for your questions, because the questions were awesome.
Thank you so much, Ann-Sophie, and see you soon. Bye-bye. Thanks.
See you soon.
Bye, everyone.
Stay safe. Bye.